Good afternoon, everyone. My name is Tabalo Abelchauke, and I'm a training and outreach coordinator at Central Node HJ Bionet based in Cape Town, South Africa. And I'm part of the HJ Bionet Seminar Coordinators Task Force. There's a group of us that make sure that on a monthly basis throughout the year, we organize different webinars based on different themes and topics, and we attract different speakers, mainly from Africa, but we also have a global audience and speaker base as well. But for now, a speaker is African, and I'm excited about it, as most of you are as well. I'm glad that you could join us for the March webinar under the theme of African genome variation. Most of us are under lockdown in most countries due to the ensuing outbreak of coronavirus. So thank you so much for joining us during these stressful times. The talk that we have today is titled, as you can see on the slides, The Opportunity in African Genome Resource for Precision Medicine. And this talk is going to be given by Dr. Segun Fatumo, who's an assistant professor of genetic epidemiology and bioinformatics at the London School of Hygiene and Tropical Medicine and MRC UV, Uganda. Just for housekeeping, this talk will be for 40 minutes, or rather 40 to 45 minutes, where the speaker is going to be presenting the research and findings. And we're going to have a 10 to 15 minute comment, question, suggestions at the end of the talk. Just to make sure that we have order, if you want to speak, you can type your question in the chat box, which is on my left side of the screen at the bottom. So you can just write down what is A or what is B if you have a question. And if it's a burning question, you can, at the top of your screen, there's an icon with a person raising their hand, and you can use that you can click on it, and we're going to see that you want to speak, and we can give you space to speak instead of just speaking over the speaker, or rather Dr. Segun Fatumo, who's our speaker for today. So instead of speaking over him, kindly type in the chat box or raise your hand using that icon that I've just showed you. I'm just going to do a quick introduction of what he's done. By the way, we sent out an ad, and I'm not going to read out his CV because he's done so many things, and I don't want to just waste time speaking about what he's done instead of actually giving him the platform to speak. Dr. Segun Fatumo is an assistant professor of genetic epidemiology and bioinformatics at the London School of Hygiene and Tropical Medicine and MRC UV, Uganda. He's also a senior scientist at HJ Bionet, but the node, which is the Center for Genomics Research and Innovation, National Biotechnology Development Agency in Abuja, Nigeria. There's other positions that he holds, and he's founded some movements in Africa that have to do with bioinformatics. But if you want to read his full CV, follow him on Twitter, email him and ask him, or Google him as well. It's very much Googleable or findable on Google. But without further ado, I would like to say thank you so much to Dr. Segun Fatumo. The mic is yours. You can go ahead and start your presentation.
Thank you very much for that kind introduction, and thanks to everyone for joining. I do hope you can hear me clearly.
So we can hear you.
Okay, so my name is Segun Fatumo. I'm currently employed at the MRC Uganda and London School of Hygiene and Tropical Medicine as an assistant professor. I'm also fully involved at the HJ Bionet node in Abuja as a senior scientist. So today, I'll be speaking on the opportunity in African genome resource for precision medicine. So I chose this topic to showcase the endless opportunity in African genome resource and to re-echo that we are now in the time for genome revolution in Africa. In this presentation, I will try very hard not to talk about the many challenges in performing genomic study in the African settings. Maybe that will be a discussion for another webinar. So what is precision medicine and why is this important? Precision medicine is an approach for disease treatment and prevention that take into consideration account of individual variability in gene, environment, and lifestyle for each person. So this approach, for example, to medicine is in contrast to the current standard one-size-fits-all approach, in which disease treatment and prevention strategies are developed for an average person. And in that case, so less consideration is given to differences in individuals. As you can see on this slide, so the big box on your left is showing the patient population, indicating that patients do have different genetic composition and they are exposed to different environments. So the standard one-size-fits-all approach assumes that all patients are the same and all patients have the same genetic composition and they are exposed to the same environment. So the same treatment A is administered to all of them, like you can see from here. The implication of this is that such treatment is not always very effective for many patients. So a tailored approach tries to group the patients and administer an appropriate treatment for each group. For example, treatment A could be appropriate for African living in Africa. Treatment B could be appropriate for African living outside Africa. Treatment C for European, treatment D for Asian, and so on and so forth. So therefore, precision medicine is an approach to detecting or treating or managing disease that is based on individual variation in genetics, environmental and the lifestyle factors. Precision medicine is expected to reduce health disparity, but this will be possible only if studies have adequate representation of racial minority. For the rest of my presentation, please permit me to use the word precision medicine and genome medicine interchangeably. Okay, so in this slide shows a review paper that was published in 2016. So this paper revealed that 96% of participants in genome-wide adaptation study, GWAS, were individuals of European descent. And 7 years down the line, in 2016, so the finding indicate that proportion of individuals included in GWAS who are not European descent has increased by 19%. But much of the increase that you see is as a result of more study being done in Asian population. The degree of which population of Africans are represented in GWAS have barely shifted, like you can see from here. The finding, the prompted warning that a much broader range of population should be investigated in order to avoid precision medicine being of benefit to only a privileged few. Even up to last year in 2019, about 79% of all GWAS participants are of European descent. And this means that using primarily white European population to draw a conclusion on precision medicine. So the implication is that this could lead to misleading and also it can also aggravate health disparity. As you may have noticed on this slide, the fraction of non-European individuals in GWAS has stagnated or declined since the late 2014. So this suggests that we are not on the track to correct the imbalance. This is especially problematic as a previous study has shown that African study contributes an outsized number of associations relative to study of similar size in European population. In this presentation, I will show you an example of such from our genomic study in Uganda and other African populations. Now, this slide represents the distribution of global population in 2017. So we must bear in mind that continental Europe represents only 10% of the world population, and all individuals of European descent worldwide only make up 16% of global population, including European descent individuals in America and all over the world. I think there's an urgent need to expand the diversity of population being studied in GWAS for a more complete and effective precision medicine for all. As you can see on this slide, governments of over a dozen countries have invested heavily in establishing national genomic medicine initiatives. From Saudi Arabia, Qatar, Turkey, even Brazil, Japan, even Estonia, with a population of only 1.3 million people. Just few months ago, the Prime Minister of the United Kingdom, Boris Johnson, announced investments of over £200 million to do whole genome sequencing of up to 500,000 individuals in the UK Biobank. So when this is complete, this will represent the world's largest genetic project to be sequenced, to be whole genome sequence. In 2016, the Chinese government announced a bold initiative to become a global superpower in precision medicine with an investment of 9.2 billion US dollars. So the Chinese initiative has three core objectives. So they are going to be focusing on recruiting millions of participants from 7 main regions of China to form a nationally represented cohort. And they will be focusing on cardiovascular disease and other diseases. So the Chinese 9.2 billion precision medicine initiative could see about 100 million whole genome sequence by 2030. Of course, many people are familiar with the African Genome Project that is tagged all of us. And the aim of this project is to enroll 1 million participants when completed by 2022. All these country-level initiatives are an important part of the global effort for precision medicine. Maybe you've already noticed that there is no known initiative from any African country yet. In case you are looking, this is the African map here. So this slide is very similar to the last slide I showed just now. This is for a precision medicine paper that was published in 2018, but the focus is on type 2 diabetes. So it was evident that most of the country level initiatives on this slide are public-private initiatives, including the UK Biobank initiative and the United States All of Us project. The only exception on this slide is the genomic aggregation project in Sweden, which is public initiative, the Estonia personalized medicine pilot project, and the Saudi Human Genome project. So all other country level initiatives that you see here, they are private public initiatives. So this gives some hope that the genomic researcher or genetic researcher within Africa may want to look beyond government funding, which may not even be available in some countries. Like you may already know, about 100,000 years ago, human beings migrated out of Africa and started to populate the globe. They carry with them part of their ancestral genetic variation. This implies that all human beings have a genetic origin in Africa. What this means literally is that everybody came out of Africa. Given the central importance of Africa to human origin and the susceptibility, there's a clear scientific and public health need to develop large-scale efforts examining diseases in Africa population. African populations are known to harbor the greatest genetic diversity, emanated from estimated 2,000 distinct languages in Africa and about 3,000 ethnic groups in Africa. And from this slide also, you can see that currently close to 50% of African population are children under 18 years old. By 2100, 50% of the world children under 18 will be living in Africa. This is really very important for precision medicine. And also, I always like to show this slide to let people know that Africa has so many countries, more than 50 countries, with 1.3 billion people living in them. In order to demonstrate the value of African genome, we recently published our work in Cell. The project represents one of the largest collection of GWAS and whole genome sequence data in Africa. So in this study, we collected and analyzed genome-wide data from 6,400 people from rural community in Uganda. We also incorporated data from other parts of Africa, including data from Nigeria, Kenya, Ghana, and South Africa, making a total of more than 14,000 individuals across Africa. We measure a range of communicable and non-communicable health indices, such as HIV, hepatitis B and C, BMI, liver function, full blood count, HbA1c, which is an indicator that is commonly used to diagnose diabetes. In the next slide, I'll be showing you some pictures from the data collection stage and the settings where the data was collected. So in this slide, I attempt to show you the procedure we took in the data collection stage. Approval of the study procedures, including material transfer agreements, were given by UBRI and also Uganda National Council of Science and Technology. So the community mobilization precedes the recruitment process and focuses on engaging with participants at both community and individual levels. So the local leaders, they are first sensitized about the study activities, and their permission we sought their permission before holding community meetings. So community mobilization was followed by mapping, then a sensor and medical survey for health, lifestyle, biophysical and blood measurements in order to ascertain disease outcome and risk factor for selected participants. So 5,000 individuals were recruited. 5,000 individuals and genetic epidemiology study. Another 2,000 individuals were recruited for whole genome sequencing. In our pilot data collection, we found a high prevalence of cardiometabolic risk factors in this cohort in Uganda. And this cohort is a cohort with an average age of less than 50. We also noted a very low awareness of the research participants. For example, none of those with high cholesterol was aware that they have high cholesterol. And about 80% of those with high blood pressure were not aware. Like you can see also from this slide, more than half of people with HbA1c, that is more than 6%, were not aware. I think this really called for a concern. So as part of this project, we gave support to the research participants. Some of them were treated for the disease that they have. Also observed in this project is comorbidity of communicable and non-communicable disease. For example, you can see on the statement, the last slide, the last statement, you see there are some research participants with abnormal liver function. They also have hepatitis B and C. So the message from this cohort is that there is relatively high prevalence of risk factor of cardiometabolic disease for a young African population, where 90% of them are less than 50 years old. 22% prevalence of hypertension is really high, and 31% prevalence of central obesity in women is very, very high for a young population. Like I said previously, I think the most worrisome is a low awareness of this condition coupled with low access to care. This is especially important bearing in mind that about 60% of Africans still live in similar rural settings, as only 40% of Africans live in urban areas. So this population actually represents what you will see in many rural African settings. Though many of these sound like a very challenging situation, but these cohorts offer a rich platform to investigate the interplay between communicable and non-communicable diseases in Africa. We now have the opportunity to create health awareness, which in turn may help us to narrow the health disparity, first of all between rural and urban communities within Africa, and also between Africa and the rest of the world. And one other opportunity is that I think Africa are still more receptive to research, as it is not too complicated to recruit people and get them motivated to participate in research compared to what you will see in other parts of the world. And these cohorts also provide opportunity to build capacity building at different levels. There were, like you saw from the previous slide, there are so many people that were recruited, training were conducted, and many people were trained on how to do this kind of study. So we genotyped the 5,000 recruited individuals on 2.5 million Illumina chip array. This array was the most dense array at that time. I think if this were to be genotyped today, it would probably be genotyped on the H3 African chip array or similar African genotyping array. Whole genome sequencing was done on the nearly 2,000 recruited individuals. So we performed stringent quality control both on the genotype and the whole genome dataset, and they were meant to get together to improve power to detect variant. After quality control and imputation, both the genotype datasets and whole genome datasets were merged. We incorporated our GWAS dataset from other African populations, and this includes the Durban diabetes cohort from South Africa and the African American diabetes mellitus, that's Hadam from the Chasrotimin group. So altogether, our genomic analysis was done on 14,126 individuals across Africa. So we discovered 10 novel genetic associations with several traits and diseases. 9 of these genetic associations are only found in Africa, and would therefore not have been found in the largest study involving European individuals or other populations. Now, for those that might be interested in the very detail of how the quality control was done, so this slide is presented to see how some samples and some variants were excluded from the slide. But for this presentation, I will not be going through the detail of how the quality control was done. As we already know, a genotype imputation can help to infer missing genotype using haplotype information from reference samples. So genotype imputation can help us also to boost power to detect in GWAS, and can also be useful for meta-analysis and also for file mapping. It is particularly important for study from Africa, where sample size are very small compared to what we see in other populations, other European or Asian populations. So in our study, our imputation was done on preface data with software called Input2. and we use African-specific merge reference panel. Following imputation, duplicates and monomorphic variants were removed as part of post-imputation quality control steps. So we demonstrate the value of African genome resource by creating African unique reference panel using dataset from our 2000 whole genome sequence individuals, and also using data from African Genome Variation Project and also the very popular 1000 genome project. As you can see on the figure on the top left, our unique imputation reference panel has performed existing imputation panel in accuracy, especially when inputting into East and South African populations. The Matra plot below further show the power of imputation to identify more genome-wide significant variants when genome-wide study was done with or without imputation. On my left here is analysis that was done without imputation, and on the right here is the analysis that was done with an imputation, and you will see that you see a very strong signal. You will see extra variants that were found in imputation, and also there's another variant found here that was not found when it was done without imputation. So this slide shows the list of novel genetic associations that were found to be associated with various cardiometabolic traits and diseases. It also shows lists of distinct associations as already known loci. As you may have noticed here on the last column of the table, the minor allelic frequency of most of the variants are either 0, meaning that they are monomorphic, or very, very rare. See some of them here are either 0. So like I said, what this means is that all these associations that are found here, they would not have not been found in other populations because they are not there, because they are monomorphic in other populations, but they are only in African population. Our discovery includes a genetic variant that causes alpha thalassemia to be associated with HbA1c, which is an indicator for diagnosis diabetes. The genetic variant is present in 22% of Africans and almost absent in European population at a minor allelic frequency of 0.5%. This particular variant is thought to have become more frequent among African population because it can prevent severe malaria. So our findings suggest that as HbA1c levels are often used to diagnose diabetes globally, it is possible that diagnosis may be misdiagnosed in up to 22% of Africans because of their genetic makeup. Our study also leveraged on lower linkage disequilibrium in African datasets. Our file mapping analysis with software called Matra resulted in a narrow, credible interval for most traits. In fact, we were able to map 16 out of 41 distinct loci at a single SNP in the credible interval. So we also resolved the previously identified association with HbA1c. So I think this is one big value for using African genome resource because it helps you to find the causal variants. As you may expect, we find a higher level of genetic diversity in the Uganda population than is seen in similar studies in the rural population. In fact, around a quarter of genetic variation identified in our Uganda dataset had not been discovered before. We found that 28.7% of SNPs discovered in Uganda whole genome sequence datasets are not found in genome aggregation database. Specifically, you can see here that we identify 9.5 million novel variants that are not present in 100 genome projects or the African Genome Variation Project or UK 10K reference panel. The richness of the Uganda genome resource also offers us the opportunity to study the extent to which genetic differences encode differences in traits and disease. In this study, we also assess the heritability of all the 34 cardiometabolic traits I mentioned earlier using mixed model approach that also model environmental correlation. We found that height, for example, as one of them, is less genetically determined in Uganda relative to what you will see in European studies. So the narrow sense heritability of height was 49% in Uganda compared to the estimate of up to 70 to 80% in the European population. So one reason for this might be environmental factors such as the kind of food or nutrients that is taken in Uganda. In contrast, the heritability estimate for LDL was significantly higher in Uganda population. On the whole, our findings highlight the usefulness of examining genetically diverse population within Africa. So we now need even more and more large, diverse study of genetic causes of disease across the whole region of Africa. So collectively, our findings will foster development of new treatments that will benefit people living in Africa as well as people of African descent living all around the world. Just to recap, African Genome Resource provides so many opportunities. One is to discover novel genetic loci that are specific to African population, which may have implications for precision medicine. As we have seen earlier, there's a greater value in using African Genome Resource for generating African unique reference panel, like I showed in the previous slide. Also, the low linkage disequilibrium in African, we can leverage on that. If you compare that to other populations, it provides us with opportunity to narrow in and find map disease loci. And of course, the African Genome Resource may help with African-specific diagnosis of disease. And I'll show the example of the HB1C. So currently, the number of sample size from Africa is relatively small. I've seen studying in the population. For example, there is GWAS of EGFR, a CKD of 1 million individuals in the urban population. But in Africa, this study is considered to be the largest genome ever. It's only about 14,000 people. So there's currently the study in Africa, the size are really still very small. So in previous slide, I demonstrate the value of genotype imputation for improving power to detect association. There are other strategies to boost power, and this includes multivariant GWAS or to recruit large sample size. So like you may already know, so GWAS analysis are generally univariant in nature. So what this means is that it focuses on a single phenotype with SNPs. However, many human traits, they are highly correlated. For example, LDL and HDL are correlated. Height and weight are correlated. Red blood cell, white blood cell are correlated. So this correlation can be leveraged on to possibly improve the power of genetic association tests to identify markers associated with one or more of those traits. So in the first ever multivariant GWAS, we use different multivariant methods to boost power, and we identify 3 known loci for the proof and 5 novel loci that were not identified using standard GWAS method. So currently, two of my MSc students are working on applying multivariance method to correlated traits. Toure on the right from Mali is exploring different African and African American data sets, including Black individuals from UK Biobank, to discover potential genetic loci for the 4 blood lipid traits using multivariance method. So Toure is currently applying this method to summarize statistics. So Brenda on the right from Nigeria will be using the same method and similar data set to explore multivariance on understanding genetic of hypertension in Africa. So Mariam from Uganda is trying to assess genetic determinants of chronic kidney disease and blood pressure in our Uganda data set. So both Marion and Chisholm on the left are doing internship in preparation for a PhD program with me. So Chisholm will explore the same method on anthropometric traits, while Marion will explore that on chronic kidney disease. So the GWAS of metabolic traits that we started a few years ago by my former student Abdullah from the University of Science and Technology in Mali. So it's now a collaborative work with some of my colleagues in South Africa and Uganda. So we're trying to apply different methods, multivariate methods to boost power to get genetic variants that are associated with metabolic traits. And here you can see some of the work that we have done so far. You can see here on top here at the table, the multivariate method that are used to boost power identify loci that are known to be associated with metabolic traits, and there are also novel loci that are not previously known to be associated with metabolic traits. Ultimately, we want to get bigger sample size to be able to boost power and detect more variants. Maybe you already know there are so many cohorts within Africa, under H3 Africa, that are working to pull data together to identify, to conduct large data sets. There's a group called Cardiovascular Issue Africa Innovation Resource Share that aims to analyze GWAS of up to 60,000 individuals. Currently, you may have heard about NCD-GHS, that's the Nigerian 100K NCD Genomic project that aims to recruit 100,000 individuals from Nigeria in order to do a genomic study of NCD in Nigeria. So this sample size, when put together, so I'm hoping that in the next 2 or 3 years, so these two consortia will come together and put data together to be able to have a study that will be comparable to what is currently available in other parts of the world. Like I said, my interest for this presentation is to actually focus on the opportunity that are available in using African genome resource. But if there are people that are actually interested in looking at the challenges of conducting genomic study in Africa, there are a lot of them which I will not be talking about them today. But the slide will be available for people to have a look at them. So in conclusion, there is a considerable progress that has been made globally in the push to implement genomic medicine as a crucial part of personalized medicine. However, the African ancestral population lags behind because of limited participation in genomic research, both as research subject and as well as research investigator. And I've shown this in one of the slides. So this limitation has resulted in a poorer evidence base for genomic medicine in diverse populations. So why there are now research that are being needed, there are now several initiatives within Africa, such as the H3 Africa that I just showed now, and the 100K genome project within Nigeria, they could help to improve data and also evidence-based to make genomic medicine useful to diverse populations. Like you saw in my presentation, our findings from a small data set of just 6,400 and 14,000 across Africa show a great potential to inform genomic medicine, not just in Africa, but globally. The dataset I've just described is available for other researchers across Africa and globally to use, and these are made available freely, and application can be made through EGA. So there's a data set on about 5,000 individual genotype on 2.5 million Illumina array. There's a low-dense 4S data set that are whole genome sequence, and also there's also three family tree data sets from Uganda that can be applied for. If anyone is interested in all these data sets, you can just apply, send an application formally to my MRC Uganda email. So there are so many people to acknowledge for this, and I will not be able to mention them one by one, particularly people that are on this paper, the same paper, and the multivariate paper that was published last year. And also for me, there are so many mentors that I've had over time for my postdoc in Sanga, which was funded by the Issue Bionet node in Abuja. And also currently, some of the work that I've described here, they are work that I've done in collaboration with my students. I have a group now that is called ATCG. That's the people that are working on anything that relates to computational genomics. So there's a lot of them. So the work that they're doing, that's the work I've been presenting here. And also, I'd like to thank the funder that funded my work currently, the NIH, and also the MU Plus in Uganda. So thank you very much. I'm happy to take any questions.
Okay, thank you so much, Segun. I saw Fozia was typing, so I'm just gonna, let me make the chat box bigger so that we can read the questions. In the chat box, she says, thanks, Fatumo. Looking at the diversity of African genotype / phenotype association in the presented work, what about specific medication and precision medicine for these specificities? Sorry, my English. There's a question from Fozia there. Segun? Hello.
Yeah, I think the purpose of precision medicine is to get treatment or diagnosis appropriate for not really for individual person, but maybe for a person. Like I showed in my presentation, that HbA1c is an indicator for diagnosis diabetes. And people, for example, that have the 22% of people that I showed, there's possibility that the way that diabetes they should be diagnosed with diabetes should be different. So that's kind of thing that precision medicine will do, not necessarily for individual person.
Okay. Someone raised their hand. Victor. Okay, let me approve Victor. Go ahead, Victor.
Yeah, thanks a lot, Segun. This was really a very nice talk. You alluded towards the end of the talk to various issues that make it difficult to expand the scope and number of enrolled patients in the African context. Could you maybe outline what you think the most important issues are and how we could move forward to increase the representation of African population and also collect more data that are relevant to precision medicine in African populations, and keeping in mind that these populations are incredibly diverse.
In my own understanding, I don't think the problem in getting individual enrolled for study in Africa, I don't think is that people are not the research participants are not available. I think that people are more receptive to research in Africa than other population of the world. Um, Maybe we just have to approach them for that. Because if you look at the response rate from European population or Asian population compared to African population, you see that people are more receptive to African population. And I understand that there is a lot of diversity across Africa, but this should be taken into consideration. So what we need, one of the things that I highlighted is that many people are trying to get funding from the government within Africa, which is not really available. Maybe people should look beyond the government and also look at public and private partnership and do the recruitment across Africa. I don't think it's difficult to recruit people across Africa.
Okay, so what you're saying is that the barriers in terms of recruiting are lower than in most parts of the world, but that funding is really the main problem. Is that the take home?
No, I'm not saying that funding is the only problem. There are so many challenges in conducting GWAS in Africa. That the problem could be a lot of problems that are highlighted on this slide and all that. But what I'm saying is that if funding is available, the recruitment of participants will not be a problem. There are challenges in recruiting people all over the world. So recruiting in Africa is not really a major problem. So if the fundings are available and the expertise also is available within Africa, so we will be able to collect a lot of samples across Africa.
Okay, thank you.
Thank you, Victor, for your question. I see someone is typing in the chat box. If you have a question, you can raise your hand as well. There's an icon. Oh, Jared Maina. Hello, great talk, Segun. How do you address the issue of population structure in your studies?
Yeah, just like what Victor said, there's so many challenges in conducting GWAS in Africa, and population structure is just one of them. And like I said at the beginning, I didn't want to emphasize so much about the challenges, but for our study, there are so many methods to cater for population structure. We use an approach called a mixed model that was implemented in the software called Java to take care of the population structure. Some of that study within Africa we use principal PCA to also to cater for population structure. So we know the problem exists, and there are approaches that you can use for that.
Jared is still typing. I'm gonna wait to see if we still have 12 minutes to go. So if you have any, okay, Jared says, I see, thank you. We still have about 11 minutes now to go. So if you have any questions or comments, for Segun, please go ahead. You can raise your hand, you can type. Nusra says, great talk, Segun. I see Faisal is typing. We're gonna wait for. Okay, Verena says, thanks, Segun. Very exciting research. Okay, a lot of people are typing now. So I'm just. Someone asks, how about the accessibility? How accessible is the data? Do you have any comment to that, Segun?
So like many other genomic data sets, so our data set is now on EGA. So that means that anybody that is using this data set for their research, they need to apply for that. And we have a data access committee that will look at your application and approve that for you. And when that is approved, so we instruct AGA to make the data set available to you. And we have both the individual level data sets available. Also, we also have the summary statistics of the GWAS that was done. We also have it available on GWAS catalog. So this can be downloaded freely without even making any application.
Okay, thank you for that. We have two more questions. One from Nusra asking about, is there any possibility of using the Uganda experience to establish a durable study in other African regions? And Faisal asks, or rather, let me let you answer that and then I'll ask what Faisal asked. So is there any possibility of using the Uganda experience to establish durable studies in other African regions?
Yeah, I think so. I think the challenges for conducting GWAS cut across so many countries. I understand that there are very specific challenges in some population, like African population. Somebody already mentioned population structure. But I think many of the method and the procedure can be applied to so many other countries.
Okay. And Faisal is asking, what is the status of integrating genomic profiling with health informatics in Africa, for example, EMR, electronic medical records, if that was unclear?
Yeah, for like many of us are aware about the UK Biobank, and they're able to incorporate genomic data set with electronic record. But maybe one problem in many countries in Africa is that the electronic record is maybe they are not available. Well, these are things that are possible. And as expertise is being developed in Africa, I think it will be easier to be able to incorporate electronic record if they're available to generate data sets.
Okay. For just saying, this is nice work. It will be helpful to implement precision medicine in Africa. So actually, by the way, we are trying to, at the Central, are trying to work on an animation for participants and for politicians or policymakers who work with precision medicine, because we've realized that for a lot of people, or rather laymen, people that don't work in bioinformatics or genomics, they don't necessarily know what pharmacogenomics is or precision medicine is. So we're trying to come up with a 2-minute, 3-minute animation that we can easily distribute on YouTube, on our social media, and just generally as well, to explain what precision medicine / pharmacogenomics is. But we're busy working on that. We have the script already. We just need to do the voiceover and the actual animation. Lilia says, great work. Did you find variants involved in rare genetic diseases in your WGS and how to deal with them? Are they communicated to participants? Segun?
Yes, I think if you find novel variants in the whole genome data set, most of them are actually rare variants. Uh, and even in other populations, the variants you found that are specific to your population are going to be rare variants. So most of the variants are actually rare variants, and that is expected.
Okay, I'm just waiting to see other people typing. Okay, leader says thanks. We still have 5 minutes, so if you have any burning questions, okay, Nusra has a follow-up. As a follow-up to your response, I think the next step would be generating research groups across Africa for for data collection. Surely a number of independent, unharmonized data are out there. So I don't know if that's a comment or a question, but it sounds like a comment. But Nusra is saying that the next step would be to generate research groups across Africa for data collection, and there must be independent and harmonized data that already exists.
I think that's what H3 Africa is doing. So there are so many groups that are doing different stuff, and they're trying to harmonize work together in order to conduct a large-scale genome study. And I will show a slide where there's a subgroup called Cardiovascular Issue Africa Innovation Resource. So this is a group of so many research groups across East Africa pulling their data sets together. And so currently they are going to put together about 60,000 samples in East Africa. But I think they do have potential of making that up to 100,000 because there are also other data sets that are not within East Africa. For example, our own Uganda data set and maybe Adam data set, DDS, and other data sets. So I think they have the potential of making up to 100,000. So like I said, in the next 2-3 years, I see potential where the Cardiovascular East African Innovative Research Group and the NCDGHS will be collaborating together and be performing the largest genome study in Africa of about 200,000 people.
You might have to put on your mentor hat now. Harun Aremu is asking, how does an early career researcher get involved in this area of research?
Yes, so like you saw, currently I have 4 students working on different projects. So we are getting them involved, we are getting them trained. Maybe you've already seen from the issue BioNet, there's going to be a training that is funded by Wellcome Trust, people to get motivated about doing genomic study in Africa. And they can start by doing an internship. Like I do have 2 students that are doing internship. They can start by doing internship maybe for 3 months or for 2 months and build their capacity. I also must say that I started doing this work under a fellowship from the HU BioNet as an early career person doing a postdoc at Shanghai Institute. So anybody that is motivated to do this work could do it.
I think that's a great answer there, Harun. So basically look out for opportunities. Ask anyone who works in this area for guidance and perhaps even for supervision wherever you are. We have 3 minutes left, and I'm just going to allow 2 more pressing questions. So if you have a question, you can raise your hand or type it in the chat box. Just so you know, we are recording this. It's going to be up on our YouTube channel with our previous webinars as well. Segun, I hope you've given consent for us to send the slides to the participants that are attending this webinar now, to send your slides with the recording as well. I hope that's okay with you. Okay, sure. Thank you. So to everybody else who requested, for those of you that are not on the consortium list, email me separately, babalo.chauke@uct.ac.za, and then I'll forward you the slides. And I'll also then send it to the consortium list with the recording. You can follow Segun on Twitter. You can email him. His email address is in the ad and it was on the slides as well. So if you want to email him to get in touch, get clarity, or if you have any other further questions, perhaps you have ideas about collaboration, you can tweet him, you can DM him, you can also email him, and that's his card name as well. All his details are on the slides. I think it's safe to say there are no more questions, and with that, I just want to say, Dr. Segun, thank you so much for an amazing talk that is very timeless and I think current, because I mean, what's happening now in the world with outbreaks and whatnot, we do know that we do need precision medicine, particularly in Africa. So thank you so much for taking time out during this stressful time to give this talk. We really appreciate it as HJ BioNet and as the task force, particularly engaging with people like you to come and give talks. So thank you so much.
Thank you very much, and thanks for giving me the opportunity.
Okay. To the attendees, thank you so much for attending. See you next month for a different speaker with a different talk. I'll communicate in due time, I think 2 weeks before or rather a week before the talk is supposed to take place in April. Enjoy your day, everybody. Thank you so much. Bye.